In this video, we will discuss on how ions or charged particles pass through the cell membrane. So if you can remember, ions, because they are charged, have difficulty passing through the cell membrane without the aid of certain proteins we call channel proteins. So in fact, they will just bounce through or bounce against the cell membrane if these proteins are absent. So indeed, there are many types of channel proteins that allow ions to go through the cell membrane. And these channel proteins vary on how they open. Okay? So first, we have the voltage-gated proteins or channel proteins. And these voltage-gated channel proteins only work if there is a change in the voltage relative on the outside and on the inside of the cell membrane. As you can see here in the first illustration, the one shown in green. So notice on top that the charge on the upper portion of the cell membrane is positive. So the ion channel is closed. But notice at the bottom, when it's open, the charge of the cell membrane on the upper portion became negative. So that change in voltage caused the channel to open. Then here on the second illustration, we have ligand-gated channels. So think of a ligand as a sort of key that will lock, have a key and lock mechanism against the channel, so that when it's attached to the channel, as shown here at the bottom portion, the channel actually opens. You can also have a sort of reversed ligand-gated channel, as shown here on the third one, wherein the lock and key mechanism is towards the inside of the cell. So on the first one, the lock and key mechanism may be only accessed from the outside. So that's what's shown there. And on the second one, the lock and key mechanism may only be accessed from the inside of the cell. Okay, and again, the keys are the ligand molecules. And here, lastly, on the right, we have an ion channel that may be opened through mechanical force. Okay, so as you can see here, the flap that is illustrated may be pushed together or pulled apart by a mechanical force. So scientists have been studying ion channels through their detection of changes in voltages across the cell membrane. So one way of them studying it is actually putting electrodes on the surfaces of cells, specifically surrounding voltage channels or ion channels. And as ions pass through the ion channel, they would detect the minute changes in the voltage, as shown here on these illustrations. So here's an example of an ion channel, a potassium channel in bacterium. So you can see here the channel is actually made of proteins, and you can see the secondary structure of the protein. You can see the alpha helices and how they are arranged. And you can see how they form a gap here in the center so that you have a channel wherein ions like potassium may pass through. So this channel is only specific for potassium. Okay, it allows potassium ions to go out of the bacterial cell, as you can see by the direction of the arrows. So the potassium ion is actually a large ion in terms of its diameter, because potassium is a large atom, is a big atom. Okay, so as it actually passes through this ion channel for potassium, it perfectly fits through the channel, and it is allowed to pass through the potassium channel. However, we have another ion present in cells called sodium ion. And the sodium ion is relatively smaller than the potassium ion. So as you can see here, as it actually passes through the channel here in this potassium ion, there are some parts of the channel wherein it cannot, it doesn't fit rightly, as shown here on the upper illustration. Okay, as it passes through the selectivity filter. Only 2 oxygen residues, for example, are able to hold it, and so it is unstable as it passes through that area of the channel. The result is it is sort of expelled back into the cytosol of the cell, meaning to say it's not allowed to continue moving through to the outside of the cell by the potassium channel. Therefore, the potassium channel is really just specific for the potassium ion, allowing the potassium ion to go out of the cell, but not other ions. And shown here, not sodium ions. Here's a voltage-gated potassium channel in eukaryotes. And this is voltage-gated because, as you can see on one of the structures, one of the subunits of this voltage-gated potassium channel, there is an alpha helix called S4, and it's magnified here. And S4 is actually voltage sensitive, so that it will only allow this voltage-gated potassium channel to open if certain changes in the voltage across the cell membrane is registered into the S4 alpha helix. So the illustration here on the right is a bit more complicated. This is now a combination of several subunits of the potassium, voltage-gated potassium channel. And you can see here, just in the center, very small, the green color-coded potassium ions passing through the channel. So here's a ribbon model of our voltage-gated potassium channel in eukaryotes. And if you compare that to the simplified drawing, which only shows one subunit, okay, we can actually remove one subunit, and we have this ribbon model showing the alpha helices. Okay. At first, it might be very difficult to reconcile how these two are the same. Okay. But in reality, okay, they are actually the same. For example, if you look at this portion of the simplified illustration, okay, this is where the pore or the channel pathway is found. It's actually located here. Okay, you can see the S5 and the S6, and then there's the potassium ion there shown in green. Okay, and the S1 here, the S2, S3, and the S4, which is sensitive to changes in voltage, is found here in this portion. Okay, and this sort of wiggly thing that is ending in an N terminus is actually this portion. So this portion here at the bottom, the beta subunit, is actually not shown on the simplified illustration. You cannot see it here. Okay, so this is a voltage-gated potassium channel because it will only open when it detects changes in voltage across the cell membrane, particularly done by the S4 alpha helix of the channel, also shown here in ribbon model. Okay, so when this S4 detects changes in the voltage, notice how it's sort of connected. You can see this connection like a bridge, S4, S5, to this portion, where this portion actually lines the pore of the channel. When this detects changes in voltage, it will actually pull There's a change in shape. This will be pulled towards that side. And this portion of the channel will open up, allowing the potassium ion to pass through. So here's a simplified illustration of how the conformational changes appear. Okay, so here's the potassium channel closed from the inside of the cell. When changes in voltage are detected, it opens up. Okay, and when potassium ions have already moved through it, it will automatically inactivate itself with the help of these inactivation peptides. They will move over the opening like this, sort of a person moving his or her palm onto his or her mouth to stop him from speaking. Okay? And so, the passage of the potassium ions will be stopped automatically in the inactivated form of the channel. And this repeats itself. So here we have facilitated diffusion, wherein we have a carrier protein this time, okay, which actually opens up and has a perfect fit with its the molecule glucose in this example. Okay, and so once glucose has bound into the carrier protein closes from the outside and opens up on the inside, and the glucose molecule is free to move into the cell. Okay, and the carrier protein repeats the process by again opening outwards, and then the glucose molecule is now able to move into the carrier protein, and the process repeats itself. So this is diffusion, but it's facilitated. So this is still not requiring any ATP. So this is passive transport from higher concentration to lower concentration. Shown here in the graph is how the presence of carrier proteins actually facilitate faster movement of solute across the concentration gradient. And then, of course, we have here active transport. So as an example, let's look at sodium. Okay? On the outside of the cell, the concentration of sodium is really high. It's 150 millimolars. Okay? While on the inside of the cell, the concentration of sodium ions is low. It's only 10 millimolars. That's 15 times greater or lesser than on the concentration on the outside. And then if you look at the potassium ion here, the concentration of the potassium ion is 5 millimolars on the outside of the cell and very big, very great, 150 millimolars on the inside of the cell. That's 28 times greater than the concentration on the outside. Yet we know the cell strives or works by moving sodium ions out of the cell and potassium ions into the cell against their concentration gradients. So we now have active transport. And this active transport particularly is performed by an enzyme, which is also a channel, called the sodium-potassium ATPase, or the sodium-potassium pump. So notice here in the illustration how the concentration of sodium is greater on the outside of the cell than on the inside. And the opposite is true for the potassium ions. So here's how the sodium-potassium ATPase works. ATP first needs to attach to the channel, causing the channel to bind with it the sodium ions. These are the sodium ions. Okay, so the attachment of ATP causes the channel to actually start closing with the sodium ions attached in it. And when ATP is hydrolyzed into ADP and P, the ATPase or the channel eventually opens to the outside, releasing the 3 sodium ions and thereby attracting 2 potassium ions in it. And then eventually it closes back on the outside. And then the potassium ions are trapped within the channel. And when the attachment of ATP, it opens back on the inside, allowing the potassium ions to go in, and the sodium ions to attach back to it, 3 sodium ions, and we go back to stage 1. So take note, this one requires ATP in order for the sodium and the potassium ions to be moved against their concentration gradients. The same mechanism happens here in the secretion of acid in the stomach. So notice there is another ATPase here, a pump. This time, a hydrogen ion and a potassium pump. So it uses ATP to move potassium and hydrogen against their concentration gradient. So potassium moves inwards, while the hydrogen ion moves outwards into the stomach, okay? Thus increasing the hydrogen ion concentration of the stomach and causing it to be acidic. So this is done by the parietal cells in the stomach. Okay, here, the parietal cells. Then we also have what we call as co-transport. So here we can see how there is an active transport happening here, wherein the cell actually moves potassium ions into the cell against the concentration gradient, and sodium ions out of the cell also against the concentration gradient. This is done by the sodium-potassium ATPase that we just learned of a while ago. And as a result of this, the sodium ion concentration on the outside of the cell is now greater than the sodium ion on the concentration on the inside of the cell. Thus, on this portion of the cell, there is a sodium ion glucose cotransporter wherein it takes advantage of the greater concentration of sodium on the outside, causing the sodium ions to just move down the concentration gradient back into the cell. But the sodium glucose cotransporter doesn't only let the sodium ion pass into the cell, it also takes advantage of that by letting a glucose molecule sort of piggyback or co-ride with the sodium ion. Thus, we call this a co-transport.